Now we're going to talk about what causes the pathology in Alzheimer's disease. It's not clearly known, but we certainly have some major hints. We begin with the protein called beta-APP, or beta-amyloid precursor protein. The APP proteins are a family that are 110 to 140 kilodaltons in molecular mass. They are in fact expressed by most tissues, especially by neurons. They reach the axon terminals and the dendrites. They're also found in some glial cells. They have a large extracellular domain, a single transmembrane domain, and a cytoplasmic side. And the pathological part of APP is thought to be a set of smaller peptides called A-beta that are released from the full-length APP by proteolytic cleavage. So there are three proteolytic cleavage sites, alpha, beta, and gamma. You can see that the A-beta peptide encompasses partially a membrane-spanning domain and partially an extracellular domain. And overproduction of A-beta 40 and A-beta 42, depending on the length, these are the lengths of the peptides, result from specific ratios of proteolytic cleavages, that is, of cleavages of the protein at the alpha, beta, and gamma sites. Here are the sites of some mutations causing familial Alzheimer's disease, just past the beta cleavage site, just past the gamma cleavage site. In fact, all of the known genetic risk factors that predispose a person to Alzheimer's disease increase the accumulation of A-beta peptides. And so on chromosome 21, which is where beta-APP lives as a gene, there are mutations, as we've seen, in A-beta itself. We've also talked about the ApoE4 polymorphism, specifically the epsilon 4 allele, which increases the density of A-beta plaques and vascular deposits. There are a couple of mutations in presenilins. There are two presenilin proteins, 1 and 2, and they too increase the production of A-beta peptides. And finally, some more recently discovered genes, such as TREM2, also increase the density of A-beta and of A-beta plaques. And so we have the amyloid hypothesis, meaning amyloid precursor protein, as a cause of Alzheimer's disease. And a recent review reminds us that the precise meaning of the amyloid hypothesis has changed over the years, and it differs among scientists. Originally, it was thought that the actual amyloid is pathogenic. More modern versions of this hypothesis actually state that A-beta and microaggregates of A-beta, not the plaques themselves, not the amyloid plaques, are the pathogenic agents. And so you could call them A-beta-derived diffusible ligands, if you like. We should also point out that we talked about tau in the last lecture. Abnormal states of tau also mediate some effects of beta-amyloid. And so a lot of the effects of beta-amyloid may come about because it makes tau accumulate or phosphorylates tau. On the other hand, this stage of tau pathology may be distal to downstream from the more toxic A-beta dimers and oligomers. When we start talking about A-beta-derived diffusible ligands, we have to ask the question, is there actually a single A-beta receptor, the way there are single receptors for many other drugs in the nervous system? And there are really no convincing data that so far suggested the presence of a single A-beta receptor. If there were one, our job would be a lot easier to understand the pathology of Alzheimer's disease. So instead, scientists have been thinking about soluble oligomers. An oligomer is an assembly of 4 to 40 peptides, all the same, all multiple copies of A beta 42. They are soluble, they don't precipitate, they interact with other proteins, and then with other proteins they precipitate to form the amyloid plaques. And indeed, soluble oligomers of A beta 42 are toxic to neurons. Unfortunately, we don't know precisely how they are toxic. The 12-mer may be the most significant toxic form, but this is not very well known. And to make things more complicated, we need to talk about the concept of cell-autonomous versus non-cell autonomous diseases. A cell-autonomous disease is one in which a cell with a mutant gene itself gets damaged, and this is the cause of the disease, or the cause of an animal's change due to a mutation. In a non-cell autonomous disease or mutation, the effect of a mutation in one cell is spread to another cell, or downstream. Now, you can say, wait a minute, all the cells in an organism have the same genes. And that's true, but there are ways of putting a cell with a specific mutated gene in an organism, in an otherwise wild-type organism, or other ways of making an insult in one cell and asking whether it's the cause of the phenotype. So, the usual assumption is that A beta 42 toxicity is a cell-autonomous process. The cell that has a bad A beta 42 dies, and that's all. But in fact, misfolded A beta 42 may spread from one cell to another. And here the example are prion diseases, in which a misfolded conformation of a prion gets spread from one cell to another and causes disease. We don't know how important this prion-like quality of A beta 42 is. We'll talk about it again when we talk about synuclein, which may also be a prion causing Parkinson's disease. So what is it about the cleavage of APP and the action of presenilin and its gamma secretase activity that is so important? Indeed, what is the function of APP in the first place? What is the function of this protein whose cleavage makes A beta 40 and A beta 42? Well, we don't know the selective advantage of APP in an organism. We do, however, know that there is another substrate, another protein, which can get cleaved by gamma secretase. So, here is the gamma secretase complex. It contains presenilin, the enzyme in it. Here is one author's view of the entire core secretase complex, containing several subunits, one of the presenilins, and a couple of other proteins with interesting names. And so what is the function of this gamma secretase complex in the first place? Well, the function, the selective advantage of having this complex, may be an entirely different substrate. It may be the Notch protein, whose cleavage is so important for developmental processes. And so some hypotheses then are that the presenilins are adventitiously or epiphenomenologically cleaving APP, but that's not their real function. The real function is in signaling like the Notch pathway. Stay tuned for this. So in terms of pathology, one could think about two types of pathology. The first is general cellular processes that could account for A beta pathology. For instance, protein homeostasis and turnover. Homeostasis means trying to keep things constant. And in this topic, we know that accumulation of erroneous, misfolded, or partially processed proteins continues as a major theme in neurodegenerative disease. So the accumulation of partially processed proteins in a neuron may be toxic. In particular, when proteins accumulate in the endoplasmic reticulum on their way to secretion but blocked from secretion, this causes a phenomenon called endoplasmic reticulum stress, or ER stress, and results in a complex set of metabolic processes termed the unfolded protein response, and an unchecked unfolded protein response can eventually cause cell death. So this is a way that erroneous, misfolded, partially processed proteins, such as the complexes between A beta and other proteins, could lead to the death of neurons. Another very general cellular process is that it might be that A beta causes ion fluxes, excessive ion fluxes, which lead to a phenomenon called excitotoxicity. This would overwhelm the cell's ability to maintain ion gradients via ion-coupled transporters. Various metabolites would accumulate in wrong compartments, extracellular or the cytosol or an organelle. And when calcium accumulates to too great an extent in the cytosol and organelles, this activates transduction systems, overactivates enzymes, and generally leads to cell death. So these are general cellular processes that could account for A beta pathology. Now, as opposed to the general cellular processes, we could think specifically about synaptic processes that cause Alzheimer's pathology. And we, over the past few decades, neuroscientists have learned so much about the function of synapses, and so much quantitative about how much transmitter is released and for how long and how long it lasts, that we have lots of good data about normal synaptic transmission. And so the hypothesis has been arisen and got some support that, in fact, either loss of synapses or subtle changes in synapses could cause the cognitive defects associated with Alzheimer's. It certainly is true that in anatomical studies, frank loss of synapses correlates better with the cognitive deficits, and these are post-mortem studies, than do plaques or tangles. But even more subtle than that is the question of whether abnormal, too little or too much synaptic transmission could cause the cognitive deficits in Alzheimer's. And so here are some possible scenarios by which soluble oligomers from A beta or plaques resulting from A beta or impaired presenilin function or calcium dysregulation could lead to altered synaptic function. So we will have to stay tuned for these specific hypotheses too. Now we're going to talk about a specific example of synaptic malfunction that might be associated with A beta peptides. It's an interesting story because it will be familiar to many neuroscientists. It involves long-term potentiation, which we abbreviate LTP. It involves slices from the hippocampus of a rat, and we've discussed the hippocampus in a previous lecture. The hippocampus is important for memory and learning, and it's one of the brain regions that degenerates in humans in Alzheimer's disease. So the top panel involves recording from cells in the hippocampus, extracellular recording, and EPSP amplitudes, excitatory postsynaptic potentials. At this point here, we give a tetanic pulse, a high-frequency pulse, to the preparation. In the normal situation, the EPSP activates and stays increased for a couple of hours. But if the medium surrounding the hippocampal slice contains A beta oligomers, then the elevated EPSP is not sustained, but it goes away after half an hour or so. The A beta oligomers can't be synthesized chemically, so instead they are synthesized in a cell line that's engineered to express A beta. Now, an example of the kind of control that one has to do in an experiment like this is to use an antibody against A beta. And so if we do the same experiment, or the experimenters, Walsh et al., do the same experiment, but now with an antibody to A beta, they see no decrease in the EPSP, showing that, in fact, it was the A beta from the conditioned medium that was the important drug. And now if we use an antibody against another molecule, a sort of control nonspecific antibody, then the A beta oligomers survive. And the trace here, the green trace, looks like the blue trace up here with the A beta oligomers. So this is a nice, elegant experiment of the several available in the literature that point to specific malfunctions in synaptic transmission and specific malfunctions that point to an inability of synapses to retain information for long periods of time. And that is, of course, part of cognition. So I've talked to you about the possible causes of Alzheimer's disease. We've talked about general cellular mechanisms for A beta toxicity. We've talked about specific synaptic mechanisms for A beta. And next time, we'll talk about models and possible therapies for Alzheimer's.